Hi there and welcome grade 12 Mindsetters. I know that I know you guys have been waiting for this time since the beginning. As you can see, I've got top 10 Mindsetter Atik
Hi
here.
everybody.
How are you, dude?
I'm well, and you?
Good. I'm so excited to have you here on the show.
It's an honor, my lady.
Thank you. Okay, so now you wanted to say a few things to those that voted, to the guys that voted you in, right?
Yes, my friends, thank you very much for all the support. Karishma, Shivani, Amanda, I can't remember all your names.
You better not leave anyone out. It's like in the
JD.
Grammys, you know, like I
It's
should
so
start
difficult
the slow
to remember the
music,
names.
slow
I'm like,
music.
oh! So I'm sorry if I forgot your name, but thank you guys. I really do appreciate all the support, and I do appreciate all the love as well.
So fantastic. So guys, hopefully you'll be seeing this face around a lot more. He will be on the Facebook page, which is
www.facebook.com/learnextra.
See, he's got it. So guys, Atik's going to be behind the scenes. He's going to be answering all of your questions. So please post them on the page. You can also chat to us on Twitter @LearnExtra. And before we start, I want to say big up to Macmillan. Thank you so much for sponsoring the show. You guys rock. We're going through solutions for all life science. So all of the content is from there. If you do have this book, you can follow with as well. But if you don't have the book, not to fear, all of the notes are on our website, and that is www.learnextra.co.za/live. This is where you need to go for all of your schedules, right?
Yes.
Your notes. And?
What?
check. I'm just trying to test him. All of your previous week's videos. Okay. So now, Atik, before we start, Aslam, I know he's like dying to start here. Tell us a little bit about yourself.
Well, I'm 19 years old. I
Newly
just want to be
19.
younger.
Yeah.
I'm 19 years old. I'm studying BCom accounting through UNISA. And I come from Durban. Ooh, yes, Durban.
Durbs. If any of you guys are from Durbs, let us know.
Oh, there's many of them. Most of my friends are tuned in.
On the page?
Yes, on the page. Well, that's about it.
Okay, fantastic. Aslam, if you have any questions that you want to ask Atik during the show, ask him about the page, even ask him a question. Apparently, you're a life science fundy, right? Okay, so this is full permission.
Oh, God.
Hey? Okay, Aslam, I think it is time to start the lesson. Take it away.
Hi, Mindsetters, welcome back. Indy's giving Atik a hard time. Yes, this poor guy is sweating. He feels like he's in the matric exam all over again, and that's how you're going to feel at the end of this year if you do not tune in regularly to us and if you do not follow up on what you have learned on a weekly basis. Guys, today we're looking at meiosis. If you're following us throughout, we started with DNA, we moved to RNA and protein synthesis last week. We looked at a few mutations. We had our own mutation in the whole mix of things where we went silent. It was a nice world for you to know what it is if you don't have sound. And we looked at frame shift and point mutations last week. We're moving on now with meiosis. Now guys, this is not a difficult chapter. This chapter is actually smaller in content than the one we've done before and the ones we're going to do afterwards. And this little lesson we can learn in about an hour.
Fantastic.
So let's go through what we have to do first. I'm glad the pen is working, everything is working so far. Let's touch wood somebody. What we're going to do today, we're going to look at, first of all, meiosis, as I've mentioned earlier. And there is some background knowledge of meiosis that you need to know. You've learned cell division in grade 10 already, and we have learned about DNA. We've learned a little bit about chromosomes, and we have spoken about haploid and diploid before. So all those words have to come back today. So we're looking at the first meiosis division, meiosis 1, and we look at prophase, metaphase, anaphase, and telophase. And we look at the second division, that is meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. So meiosis 1 and meiosis 2. Thereafter, we look at the importance of meiosis. In other words, guys, importance of meiosis meaning, why does meiosis take place? Why do we want to know about meiosis? So that's what we're looking at there. And obviously, just like in protein synthesis, just like in DNA replication, some things could go wrong. And if these things go wrong, what are the consequences? And the two examples we look at are Down syndrome and polyploidy. We'll talk about that more later. And lastly, we would want to compare what you did in grade 10, mitosis, with what we are doing now, currently, meiosis. So in a nutshell, on this one page, this is what you need to know for this section. Guys, what I write here on what we're going to discuss comes directly from the exam guideline. So in other words, what's written here is what you need to know. The textbook may have much more than what you need to know, but this is what you need to know to write an exam. First, learn what you need for the exam, and then move on to knowing more. It's nice to know more, but first know what you need to know for the exam. Good. We move straight into the lesson itself, and we start off with some terminology today. First of all, what is cell division? Cell division is a process where cells divide or reproduce. And we look at two types, that is mitosis and meiosis. The cells are the only things that actually Atik and Indy divide to multiply.
Remember, test him, not me.
No, that wasn't a question, it was a statement. That's why I addressed both of you.
Oh, thank
Okay. Chromosomes are cell parts, and we learned about chromosomes when we started with DNA and we moved on, that determine what traits a living thing will have. In other words, what we said earlier, chromosomes are made up of two chromatids. Chromatids are a strand, one strand on the set of chromosomes. And we also went further to say that a chromatid is made up of DNA. So chromosome made up of two chromatids. The chromatids are made up of long strands of DNA. We spoke about this before. Obviously, after replication, a chromosome is made up of two identical chromatids. What are daughter cells? Daughter cells are the cells that are produced at the end of cell division. In other words, the result of cell division. Cytokinesis, cyto coming from cytoplasm, and kinesis, division. So cytokinesis is a division of the cytoplasm, when the cytoplasm is split in two. In an animal cell, this is done by invagination of the cell membrane. In a plant cell, a cell plate will divide the two. We're moving on a little bit more in terms of terminology. Karyotype. Karyo coming from nucleus, type, the type of nucleus. But actually what we are talking about is how are the chromosomes arranged in the cell of this organism. So a karyotype diagram is a diagram of arrangement of chromosomes. Some books call it a karyogram instead of a karyotype diagram. Okay? First of all, we must understand that chromosomes are paired according to size and obviously according to the characteristics that they code for. We cannot have a chromosome that is coding for eye color right next to a chromosome that is coding for bum size. To put it crudely, Indy, I see you cringing there.
The bum size. I was just wondering.
More
if you're gonna start
exercise,
talking
more
about my
exercise.
bum. And you went there. And you went there.
Okay, so the arrangement is called a karyogram, and the set of chromosomes is called the karyotype. The human karyogram, we like to look at that one because it's about us. So the human karyogram shows the 23 pairs of chromosomes. Because we have 46, these 46 chromosomes we have are not just 46 any chromosomes, they are 23 pairs. One set of maternal origin we got from our mother, and the other we get from our father, maternal, paternal. The diagram here clearly shows you a male, normal male, and a normal female. Notice that the chromosomes that are lined up there, they usually start in a karyogram from the largest to the smallest, going down that way. And there you have them numbered 1, 2, 3, 4, 5, 10, 11, 12, up to number 23, which is not numbered. It's written there X, Y, and in this one it's written XX. Now sometimes in the exams or in study guides, etc., they do not have this there, the XX and the XY. How would you know the difference? And size does matter here. In the one on the right, female, the two strands are identical in size. And in the male, the one is larger or longer and the other one is shorter. So you would know the one that has different bands or strands next to each other, that's the male. And the one that have identical strands next to each other, that would be the female. The other thing we can also look at is that in every set of chromosomes here, there must be only two. So they must be paired. There you have all their paired except for this one here. So if there's something different, this one here, number 21, has 3 instead of 2. There must be something not right there. So we need to look at that. And when we do that section a little later, we talk more about it. A little bit more about the haploid-diploid story. Remember we said that our normal body cells or somatic cells have 46 chromosomes. Now here you're having a fertilized egg cell that has that. Now I would rather take that out there because you're going to get confused with somatic and zygote. So let's just leave that zygote there with 46. This is the fusion of the sperm and the egg. Where did the sperm and the egg come from? They came from germinal epithelial cells, which are 2N or diploid. One from the mother, one from the father. And when these two come together, you're getting 46 again. So the zygote that develops from there, a fertilized egg cell zygote, will have the full complement of chromosomes. Okay? If this didn't happen, then we would have had 46 plus 46, and you would get something with 92 chromosomes. Now the thing about chromosome number is that it is specific to specific species. Mind the pun on all those specific specificities. All right? In that case, what happens is, obviously there's something wrong if something has that there. And by the way, just as a matter of interest, the number of chromosomes does not mean some organism is more complex or less complex. For example, we have 46 in the 46 chromosomes in our ordinary body cells.
Everyone has 46.
Everyone has 46 unless there's a problem, like Down syndrome. It's a different story.
Okay.
The fern, on the other hand, what Llewellyn was talking about, the fern, moss is going to lead up to the fern eventually. The fern has 300 chromosomes. Now, we thought we were advanced to the fern. Now, in terms of chromosome number, there's no...
What does that mean with the difference? That the fern, I've heard things about the fern.
There's no difference in terms of anything. That is why we're mentioning it here, that the chromosome number is just specific to a particular species. That species, when it procreates, it must ensure that in that species, we must have the same chromosome number. And that is where our friend meiosis comes in. All right. Now, you did mitosis in grade 10, and we're going to go through very quickly those stages because we're going to come back to them later. The first phase is interphase, and if you remember, in interphase, the main story there was DNA replication took place. Interphase, between two phases, interphases. So when that happens, the chromatids were single, and they duplicated themselves, and they became chromosomes. The next phase that follows is prophase. And during prophase, the chromosomes become visible. There's a lot of other things that happen. The spindle fiber forms, the centrioles move to opposite poles in animal cells, and so on. But the main thing is that the chromosomes become visible. In metaphase, middle, the chromosomes are in the middle, at the middle of the cell. In anaphase of mitosis, the centromeres split, the chromatids move to opposite poles. They start moving, rather. And at telophase, we have two daughter cells that look identical to the mother cell, if you want to call it that. Identical. Mitosis, exact duplication. It's like putting a page on the photocopier. You do not expect any distortion. You expect the same copy to come out on the other one. So mitosis is exact duplication. Guys, a lot of terminology there, but the terminology is important to understand before we talk about meiosis. So with that terminology, I think you need a break.
Okay, I think it's time for a break. Let me have a look here. Cabello says, Indy, you learning, huh? By the way, you haven't given the Cheetos out yet. Yes, we did, Cabello. Yesterday we had a winner. It's all about the random acts of kindness. This is exactly what this is. We have been sponsored by Simba and Cheetos. We've got this amazing pack of all these amazing chips that we're giving away. We've even got a Mindset t-shirt we're giving away. We've got a Cheetos little notebook, and we've got all of these amazing love CDs. I think we've got the Only Love Songs album. We've got Malik, Music and Love. We've got Kaya for You. We've got Unati, I think, With Love. And it's Donald, Train of Love. It's all about love this month, guys. So what you need to do is you need to find our events page, Random Acts of Kindness. Post your picture or video on the page of you paying it forward. It doesn't take money. It only takes a little bit of time. And, hey, what would you say about Random Acts of Kindness?
Well, it's the little things that count.
Exactly.
Always the little things.
I couldn't have said it any better. Okay, guys, so now Atik is not going to be here when we get back. So he's saying goodbye. So please, please make sure that you post questions on the page for him and he will be answering because he is a life science fundy. So let's test him. Let's test him. We'll see you guys now after the break.
Bye.
Bye. Hey guys, and welcome back to Learn Extra Live. It is grade 12 life science, and I know that you grade 12s have been waiting for this, and I'm sure you guys have been watching grade 10 and 11, right? I knew that. So now, obviously, and if you guys didn't know, the show is sponsored by Macmillan, and this is this really awesome book, Solutions for All Life Science. So if you do have this book, you can follow along as Aslam is going through. All of the content is from here. Please post your questions on the page, guys. It's very, very important. There's no such thing as a silly question, only the one asked after the exam, because then it really is too late. If you see a question on the page that you understand and you feel like you can answer, please answer it, because sometimes you actually learn faster by helping each other out, right, Aslam?
Definitely.
Hey, so I really, really do say share, help.
Definitely. Thanks, Indy. Welcome back, guys. I'm hoping you're refreshed and ready to move. And what more important can Indy tell you all day today except that last statement, that you must help each other. And by helping, you're going to learn things. Even if your answer is wrong, somebody may correct you. And when they correct you, then you have understood what you have misunderstood before that. So they're going to clear up your misconception. If you keep quiet and you keep that wrong answer, you're never going to know it's wrong till the guy marks it at the end of the year. That's too late, because then there's no corrections thereafter. There's only marks. Guys, we went through very quickly mitosis before this. Remember the phases very carefully again: interphase, prophase, metaphase, anaphase, telophase. So that's where we were at. We now look at where we are now, and that is meiosis itself. So what we're trying to show here on this slide is very simple. We're saying, how does meiosis form haploid cells? So they're looking at a parent cell, which is diploid. There you go, diploid. It's just showing you only 2 chromosomes here. Okay? If that has the 2 chromosomes, this is what's happening. There's the 2 chromosomes here. In prophase, they're doing something. We're going to talk more into detail about that later. Right now, we just want to know, how does the diploid become haploid? So let's focus on that first. So when that happens, eventually after this crossing over is taking place here, you find there were 2 here, and each of these cells have 1 chromosome. So 2 became 1. In other words, it has become half. Diploid is the full set. Haploid would be half. Hap for half there. Diploid, double, but not really being double, but being the full complement. But we use the word 2n and n for that. So understand what we mean by diploid and haploid. Okay? And understand also that what is happening here is the formation of gametes, the sex cells. In the males, in humans, we're talking of sperms, and in the females, we're talking about eggs. And when this happens, this is what is happening. Each one has half of the original content. And that's what we're having there. So that's the first thing we need to understand. How does diploid change into haploid? At this stage, by the way, I just remembered that John sent me an interesting email from Mr. M.T. Aziz. I want to hats off to Mr. Aziz, was my lecturer in botany and zoology at the Transvaal College of Education. He was watching last week, Indy. He watched you and I, and he was impressed.
Fantastic. That's what I want to
hear. His
hear.
daughter wanted to watch Life Sciences in matric this year, and she insisted her father comes to watch this exciting program. So we're doing something right.
Yes, that's
I
what I
think
like
it's
to
more
hear.
you than me.
Never, never. I think you give me too much credit. It's definitely you.
No, no, no.
It's definitely the beard. The beard.
I'm too old. They're looking. All eyes are glued on you. Could be Belinda too, Indy.
I think it's Belinda actually.
She's
Belinda
in charge
is
of
our
the whole
director.
shoot, so
Yeah, exactly.
nothing goes on without her. There we go. Now, guys, we want to go into the details of what is happening. There we have our parent cell there, diploid parent cell. The first division, prophase. What has happened here? You can see clearly the centrioles forming, centrioles moving to opposite poles, and the spindle fiber forming, the nuclear membrane disintegrating. The chromatin network, which was just like a ball of wool, now becomes clearer as chromosomes. So they contracted, they become like chromosomes. And watch what is happening in this particular stage. In mitosis, we did not mention this. The chromatin became chromosomes. But the homologous pairs need not, in mitosis, lie next to each other. However, in meiosis, it's important that they lie next to each other. And the slide before this, if you remember, we saw this there. This process takes place. What is this process known as? It's known as crossing over. What is crossing over, you ask? Is when the chromatids of adjacent homologous pairs cross over, or homologs, I should say. Homologs cross over. When they touch, this point is called the chiasma. What happens here, guys, is that parts of the one are exchanged with parts of the other one. And when this happens, there's a change in the chromosome structure. And this brings about genetic variety. So let's go back to where we were. So prophase again, all of the normal stuff that happened in mitosis took place. In addition to this, two important things. Homologous chromosomes lie next to each other, and crossing over takes place. Crossing over brings about genetic variety. All right? Then we move to metaphase. Meta, just like in mitosis, the chromosomes line up at the equator in the middle of the cell. However, in meiosis, the homologous chromosomes are lying next to each other, whereas in mitosis, they were lying apart. In addition to that, in mitosis, there were one chromosome per spindle fiber. Here you can see there are two chromosomes per spindle fiber. What follows next is anaphase 1. And during anaphase 1, no centromere, nothing to do with the centromere. The chromosomes, not chromatids, chromosomes, move towards opposite poles, as you can see there. They're moving towards opposite. So half the chromosomes are moving to one pole, and the other half is moving to the other pole. Can you see that this is what brings about the halving of the chromosomes? So already in anaphase 1, the chromosome number has been reduced to half. Also in prophase 1, crossing over took place, and this brought about variety. So two important concepts here. One is the variety, and two is halving of the chromosomes, both taking place in the first phase already. First stage, I would say. Moving to telophase, in an animal cell, invagination will take place. What does that mean? Invagination, moving in of the cell membrane, pinching in of the cell membrane, whatever you want to call it. But it's an inward movement of the cell membrane. Eventually, you can see what happens. At late telophase, we have two cells from the one cell. But these are two different cells. This is also important, because in mitosis, we spoke about exact duplication. We then move straight into, there's no interphase here, we move straight to prophase 2, where we can see the chromosomes. They do not need to contract, they were already contracted in telophase 1. all the other structures are still there, the spindle fiber forms again, and the chromosomes now move to the equator in metaphase 2. Notice here they are lying singly. I want to ask you a question just now. I'll hold on to that. In anaphase 2, the chromatids separate. Why? Because the centromere splits, and the chromatids move to opposite poles. In meiosis 1, we spoke about chromosomes moving to opposite poles. Here we are talking about chromatids. What does this remind you of? Singly lying at the equator, chromatids moving to opposite poles. Yes, you're right, guys. It reminds you of mitosis. So the second meiotic division is like an ordinary mitotic division. Keep that in mind. And they move to opposite poles at anaphase 2, and finally we have 4 cells that have formed, because each one forms 2. There's 2 on this side, there's 2 on that side. 4 haploid daughter cells. Haploid because the chromosome number has been halved, and they are different. If you could look at them, there's a change. There's a slight blue in that pink. We didn't have it before. Why? Because crossing over took place in the division 1. That, guys, is the whole of meiosis in diagrams. I'm going to now take time to show you the same process in videos. So I'm going to get a rest on my voice as well, Indy.
But we so love listening to you.
Ah, that's not good, that. I hope I know what I'm doing.
Okay, well, you know what I'm going to do? While you're trying to figure out those, I get to have my little spot and have a little chat. Guys, please make sure that you have downloaded these notes, because if you can work along with us, you can keep those notes, you can share it with your friends, you can have study groups, and you can help each other out. If you haven't downloaded them yet, it's learnextra.co.za/live. You can get last week's notes and the week before, and going forward, it's going to be, this is the best URL to have. This is the place where you need to go to find everything you need. You've got schedules there as well as last week's videos. It's pretty awesome. So now I think Aslam's got it. Are you nearly ready for me?
I'm really
not
at all.
He's still there. I can just see everything moving up and down. But you know what, guys? I think we're going to take a little ad break. Are you okay with that, Aslam?
Yeah.
Okay. Guys,
I'm
I see there's lots and
let's
lots
take a
of
break.
photos on the page. Oh, what am I saying? Photos, questions. Please keep them coming. I'm going to get Aslam to have a look at them during the break. And what we'll do is we'll leave the last 10 minutes, well, I'm hoping, so we can answer your guys' questions. See you now after the break. Hi guys, and welcome back to Life Science Grade 12s. I hope that you are riveted. I know that you are, because there are so many posts on the page. I'm really trying to keep up. Mosa, Memory, Caroline, Camogelo, Monty, Cabello. Cabello is making me laugh the whole time. He's so funny. Let's have a look there. Ziabio, Camogelo, Yolanda. Guys, what we're going to do is we're going to go through all of your questions in the last 10 minutes of the show. Keep sending them. I'll compile them for Aslam, and we'll see how many we can get in the last 10 minutes. But I'm not going to waste any more time, and I'm going to pass it straight back to Aslam. Aslam, take it away.
Thank you, Indy. There we go, guys. Technology on our side now. The first phase is prophase. Please watch carefully what's happening to the chromosomes. I will stop when I have to. Notice the chromosomes becoming visible. And what's very important is that homologous chromosomes, I can't write on that, sorry, homologous chromosomes lying next to each other. This is the highlight of prophase 1 in meiosis as opposed to mitosis, because everything else that happened in mitosis will take place here. But this, and watch what happens after. This is very interesting to watch what happens after this. If after this you don't understand what is meant by crossing over and the significance, then I don't know. Just watch what happens to the chromosomes as we go on. They are lying next to each other, the same size with the same size as usual. All right. where they are touching, that's called synapses. You don't have to know that for exams. What is more important is what they're going to show now. Watch this very carefully. You got it? You got that? They were lying next to each other, different colors, and some parts of this guy went there and some parts of this guy went there. This is what crossing over means. There's an exchange of genetic material. There's an exchange of DNA. And therefore, the chromosomes that are resultant after this process are not the same as they were before. This is what brings about a variation. Right? That's the first thing. So that was crossing over. And we move on to metaphase 1, the second stage of meiosis. And this is very short. They are lying at the equator such that one there. And this is a comparison to mitosis. Notice in mitosis, they are lying singly. In meiosis, they were lying together. Good. Anaphase 1. John did something for me, but it doesn't seem to work. But anyway, we'll watch what happens. Right? Anaphase 1, there we go. Watch what's happening. The chromosomes separate and they move towards opposite poles. The chromosomes moving. Guys, we only put this here to show you the movement so it stays in your mind. In mitosis, that's what would happen. The chromatids would move to opposite poles. In telophase 1, this is what's happening. Watch carefully. There are 2 cells with half the chromosome number. We started with 4, they've got 2. All right, let me go back there. Prophase 2. That works for me. There you go, prophase 2. Same story. The chromosomes are there. The homologous are not lying next to each other. But everything else, spindle fiber has formed. The chromosomes are visible. They were obviously all visible already before this. Right, you got the idea there. So prophase 2 would be followed by metaphase 2. Not working very well for me, darling. Let's just see what happens there. Okay, carry on there. There's your metaphase 2. Watch what happens there. They are lying at the equator, however, singly. All right? That's all you need to know there. And we move to anaphase 2. And there we go. Watch what has happened. The centromere has split, and the chromatids are moving to opposite poles. The chromatids are moving to opposite poles, not the chromosomes. In anaphase 1, the chromosomes were moving to opposite poles. All right. Go back there. And we have telophase 2, the last one. That's the last one, guys. Watch what happens here now. Same invagination takes place. The chromosomes are at opposite poles. They go back into the chromatin network. We have 4 cells that have developed here. Notice in mitosis, 2 cells develop at the end. In meiosis, 4 cells. Why 4 cells, you ask? Because the division takes place twice. There are stage 1 meiosis, stage 2. 1 cell divided to form 2, and the 2 cells divided again to form 4. And that is why we have 4 cells at the end there. That's the end there. There was a story with the sperm and the egg again. There's your haploid sperm. Remember, people, this is just a diagram and it's showing you only 2 chromosomes. In reality, if it's a human sperm, it should have, that's it, 23 chromosomes, and there should be 23 in the egg. And when they fuse, then the diploid number is restored. I'm hoping that makes it easier for you. because we put in some animation so you can actually see what is happening. We're not just talking and giving you signs. As you know, one of my teachers at school says, you either teach science or you teach science. We don't want to teach you science.
Science.
We want to teach you science. So that is why we put that little video so you can see what is happening. For me, I believe when you see what is happening, Indy, it stays longer in the mind.
It's so true. It helps me so much. And Cabello, I had to read it. I love Cabello's posts. He says, let's take it, Aslam. So everyone is very, very impressed. Takalani says, wow, technology. Kamogelo says, wow, that video stuff is cool. Wow, you guys are the best. So they are loving it.
Oh,
And
they're
I
loving
love
it.
those
Good.
animations.
Good, that's good. The idea is that you must understand things better. And there's another diagram just to show you what we meant by the homologous chromosomes lying next to each other. When this crossing over takes place, this is called the chiasma for one, and if we make that plural, chiasmata, if there's more than one. Notice what's happening. Blue-brown became blue and brown and blue. So what has happened? Something from here has gone there, and something from there has come here. And therefore, the resultant chromosomes are not the same anymore, and this brings about variety. The other thing that causes variety is when these chromosomes in metaphase are lying at the equator. There's no guarantee which chromosome is going to lie where. Remember, we have 46 such chromosomes. So the way they lie, there's a lot of variety in which they can change their position. And because of this, when they split, there's no guarantee where they will go. So in reality, it means that there's no guarantee which sperm, or egg, would get which chromosome. And this becomes random. Just like the lotto. If we knew what the lotto numbers were supposed to be for tonight, or Saturday, maybe we wouldn't be sitting here today.
If only.
We would be already giving that out, and we'd be all winning the money there. So the same way, the randomness. Why? Because nobody knows where the different balls will go. In the same way, here, we don't know which chromosome goes to which sperm or egg. And in that way, it gives more variety. So one variety came from crossing over. The second variety came from the random assortment of the chromosomes, because they are positioned randomly during metaphase at the equator. Now, why is this process important for us? By now, you should pick up those bits as we were going along. Why does meiosis take place? The first one, obvious, was that it reduces the number of chromosomes by half. Now, you may ask, so what? What's so important about this? Because it reduces the chromosome number by half, it allows the maintenance of the chromosome number from generation, not the program you're watching on TV, to generation. In other words, from one generation to the next generation, from parents to offspring, the chromosome number remains the same. So humans will always have 46 chromosomes, unless something goes wrong, which we'll talk about later. Further, the formation of gametes. Obviously, this is also important, because if gametes were not formed, there would be no fertilization, there would be no offspring, no good. Also ensures that that we've already encompassed there, ensures that the chromosome number stays the same over generations. And lastly, it also is important to introduce genetic variation. And guys, this is one of the most important things about sexual reproduction. Sexual reproduction brings about variety in the species, because there are two individuals that are involved, and they are bringing their chromosomes together, and these chromosomes will determine the outcome of the offspring. Whereas in asexual reproduction, it's one individual giving rise to more of the same, almost like making a clone of oneself. So that's the difference between asexual and sexual. So meiosis plays an important role in bringing about this variety that we are talking about. One is obviously it's two different people or two different organisms coming together. That's already a difference. Furthermore, while meiosis was taking place, there was this crossing over that took place. That brings more difference. Thirdly, during metaphase, the random assortment of the chromosomes, and we don't know where they're going to go, makes another difference for this whole process. The fact that fertilization then is a different story on its own, that we don't know which sperm which of the 100 or 300, 100 to 300 million sperms will fertilize the one egg. We don't know which one is going to do that. And at the same time, we don't know which egg is going to fertilize, when, which month, which day. We don't know all this. So those things all together bring about what we call genetic variation in species. We're going to talk much more about that in evolution when we get there. Are we together there on the page, guys? We want to move one step further. And
Should we go for a little break?
You want to?
And I'll tell you exactly why. Because Meiosis says to me on the page, what happens when meiosis goes wrong? No crossover happens. And if you guys have the notes, then you know what's coming up. So we'll see you now after the break. Hi guys, and welcome back. This week, literally last 10 minutes of the show, Matrix, I can't believe how fast this has gone. We have all of your questions. We're going to leave the last 5 minutes to take them because Aslam has to go through this very, very important work. But keep those questions coming. I am watching the page, and thank you so much for having such an amazing time on the page with me today, guys. The page is on fire, Aslam. You have no idea. I'm finding it very difficult to keep up.
I can feel the heat right here.
Can feel the heat.
Guys, welcome back. I'm hoping that you carried out with us this journey of being a chromosome. Try this in school when you have time. Get the whole class together or get groups together and act as chromosomes and act out what will happen during meiosis. You will never forget that, surely. And if you do that, obviously, please do not try to separate the centromeres and the things like that. It's only the first stage of meiosis you should do as a play, not the rest of it. Otherwise, you're going to start suing me for tearing somebody's legs apart. What happens when meiosis goes wrong? Sometimes meiosis, obviously, they may be faulty. One of the pairs of chromosomes does not separate when they move to the poles. Usually, during the first meiotic division, some textbooks say during the first anaphase or the second anaphase. It doesn't matter whether it's the first anaphase or the second. Remember, you do not have to know the names of the phase. We use the words of the phase here, the names of the phase, so that you can just get an understanding of what happens. You just need to know the stage 1 and stage 2 and what happens, the sequence of events. So if during the so-called anaphase 1 or 2, the chromosomes or the chromatids do not separate, then in this case, one of the gametes, usually the ovum, then ends up with an extra chromosome. Remember the slide we had first when chromosome number 21 had 3 instead of 2? That means this child that was born from there had an extra chromosome number 21. It's called trisomy 21. Okay? And what happens because of that? This child suffers from Down syndrome. And a child or a human being that suffers from Down syndrome has some definite characteristics. The small oblique eyes, the flattened nasal bridge, the epicanthic fold, that's the slanted eye or a fold on the eyelid there as such, small underdeveloped ears set low on the head, down there. And also you'll see the mouth is usually more than often open. Protruding tongue, and the neck area is broad. There are other difficulties that this individual may suffer from. Those are some of the symptoms of Down syndrome that we need to know. Now, that is just one chromosome that has doubled, or rather, the child or the human being or the offspring or the organism has one extra chromosome. That is known as Down syndrome or trisomy. It's also known as aneuploidy, where just one chromosome has duplicated in that way. Sometimes, however, the entire set of chromosomes do not separate. So therefore, one sperm or egg, whatever, has an extra set of chromosomes. In other words, it has the diploid instead of the haploid, the 2N instead of the N. So the gamete has 2N. Some have extra sets of chromosomes in the cells. Organisms with 3 or more sets of chromosomes are called polyploids. Now here's a simple diagram to show you how that happens. Here's the haploid, here's the diploid. What has happened is, haploid is single chromatids there, or some chromosomes rather. There's diploid, the homologous pairs are there. Now what happens if this guy has 3 of each? It's triploid. If he has 4 of each, tetraploid. If he has 8, it's octaploid. But anything above 3, triploid and above, anything 3 and more is known as polyploid. Now, polyploidy is more prevalent among plants than in animals. Generally, animals that have more than one set of chromosomes do not survive. However, recent studies have found that in certain ponds in America, they use carp fish, which are polyploid. And because they are polyploid, they are sterile. They do not give rise to more carp. They found this useful to get rid of plant material that was over-flooding or over-growing in the lake there. But in a plant's family, this polyploidy seems to be an advantage. For example, take this delicious strawberries that we see on the board. And if you look at the one on the left, Indy, and the one on the right, what's the main difference, Indy?
One is much bigger.
Good enough. Don't even think further.
The seeds are bigger on the one,
okay.
as well as the top, what do you call that? The greenery. The greenery is definitely bigger.
Right. Okay, so what you're saying that this one is larger. Okay? That's the most important difference here. And in this particular case, that's the truth. The strawberries that we eat are normally octaploid, Indy. That means they have 8 sets of chromosomes. And they are larger because of that. So the industry uses this to get better fruit, longer lasting fruit, fruit that is more resistant to diseases, etc. Seedless bananas, for example. Seedless bananas, for example, is also one of them. I think our diagram just disappeared on us there. The bananas disappeared, okay. All right. Question time, Indy.
Okay, sure. I have so many on the page here. Let's start straight from the beginning. Anele asks, during metaphase 1, do the chromosome F maternal origin necessarily on one side with all those F paternal origin? Did that make sense?
So what she's trying to say is whether the maternal chromosomes are separate from the paternal chromosomes. That's true. The paternal chromosomes will be on the one side and the maternal will be on the other side. But don't stress about that because it's not important for the exam.
Okay, fantastic. So Nele, you heard that from Aslam. Okay, so Fisor says, why is the second stage of meiosis similar to mitosis?
Why? The reason for it is simple, because the chromosome number is already halved, so we do not need to halve the chromosome number. Remember, it's similar, it's not the same. It follows the same pattern. And what happens is the anaphase, the metaphase especially, are very similar to mitosis because they are lying at the equator. So I'm giving you in which way it is similar, because that's what you should be asking. In which way they're similar. In metaphase, they're lying at the equator, but singly. And in anaphase, the centromere splits, and the chromatids move to opposite poles.
Fantastic. Okay, so now this one is from Simanga, and Simanga asks, what is the importance for the chromosomes to line up on the equator in the metaphase 2?
Okay, that's the importance. Again, a question that shouldn't be in the exams as such, but why are they there? Remember, this is also just understand one important thing. There is no such thing as prophase, metaphase, anaphase, telophase in a cell. All these things happen like that, in a click of a finger. We have given them names so that it's easier for us to understand them. They are in that area, first of all, in metaphase 1, let's go to that. In such a way, the significance of metaphase 1 is more important here. Metaphase 1, when they're lining there, they're lying in a random, not the fact that they're in the middle, that's not that important, but the fact that they're lying there in a random fashion brings about variety. In the second phase, it's the same, they're still lying randomly, and therefore when the chromatids separate, and they move to opposite poles, we don't know which chromatid is going to get to which sperm or egg eventually. So the important thing is how they're lying there. The randomness is more important than the fact that they're lying in the middle.
Okay, fantastic. Felicity asks, she says, what are the main purposes of DNA replication and crossing over?
Okay, let's start with DNA replication. DNA replication, remember at the end of meiosis and mitosis, the chromatids are single-stranded. So during interphase, the DNA makes a copy of itself so that that chromatid gets a partner and is now a chromosome. So each chromatid must have a mate next to it, and that will form the whole chromosome. That's the significance of replication. What was the second one was? Of crossing over.
Yes.
Crossing over, and you listen to this very carefully. I'm going to say it only one time, and I'm going to say it slowly. Crossing over allows for genetic exchange, or the exchange of genes. And when the genes are exchanged, this brings about variety. So crossing over allows for the exchange of genes, which brings about variety.
Fantastic. Can you believe that our producer didn't think that we had enough questions? We could actually fill an entire show, right?
We could definitely just have one more show for tonight, right till midnight.
Exactly. Okay, before I go, guys, I want to say a big thank you to Aslam. The mindsetters on the page, Aslam, really have been so thankful to you, and they said that you are the best science teacher, and life science teacher, and they said that they would actually love to have you
That means
as
they've
their
only
life
seen
science
one
teacher.
life science teacher so far. That's what they say.
No, lies. They've seen the other two before. Shame, I shouldn't say that. They might get upset.
No, for
Llewellyn.
grade 12. for
Llewellyn.
grade 12, okay.
Grade 12s. He can be your life science teacher. All you need to do is tune into 319 to learn more and learn extra. Thank you so much, Life Sciences. Grade 12s, thank you for tuning in. It has been amazing. Macmillan, thank you so much for sponsoring the show and making this all possible. I've had such a fun time with you on the Facebook page, and that's facebook.com/learn extra, and on Twitter @learn extra. Thank you to all the mindsetters who have also helped each other out. You know who you all are. And this is me, Indiana, bidding you farewell. Have a good evening, guys. See you same time, same place next week. Bye.
Enjoy.